Great. Thank you very much, Jessica. Hi, everyone. Thank you for being here. Welcome to this session of the International Day of Women in Statistics and Data Science on Junior Advances in Bayesian Methods for Complex Data. This session is sponsored by j-ISBA, which is the junior section of ISBA, the International Society for Bayesian Analysis. j-ISBA is dedicated to students and early career researchers and aims to provide platforms for academic networking for early career researchers and to stimulate connections within the larger Bayesian community. My name is Cecilia Balocchi. I am a lecturer in statistics at the University of Edinburgh, and I'm fortunate to serve as a board member of j-ISBA. Today's session will spotlight the work of 4 members of our community: Sally Paganin, Margarita Groshanina, Maria Mamajiwala, and Maria Vladimirova. Now we're going to start with Sally. Sally is an assistant professor in the Department of Statistics at the Ohio State University. Her research focuses on developing Bayesian methods and models for the analysis of complex data and has a focus on Bayesian nonparametrics, model for latent variables, and model assessment. Sally, please, you can start sharing your screen and telling more about.
Of course. Now you should see everything. Right.
Yes, perfect.
Fantastic. So many thanks, Cecilia, for the introduction and for organizing this section, the session today here, and also thank j-ISBA and the Caucus of Women in Statistics for organizing the conference today. Today I'm going to talk about a Bayesian framework for early cancer screening, which is a joint work with Jeff Miller, who is an associate professor at Harvard Biostatistics. And as the title already says, this work is motivated by a really cool problem in public health, which is the development of tools for cancer screening and characterization. And there is a huge focus on developing tools that are non-invasive. And one of the most promising technologies is based on analyzing blood samples, which are routinely collected in our lifetime. It is possible to use blood samples for cancer detection or characterization because it has been discovered that blood sample contains something called self-floating DNA, which is literally fragments of DNA that float in the blood and that is released by cells during their lifetime. And the rationale to use these technologies for cancer detection is in that if you're developing a cancer mouse, you will have some cancer cells containing some DNA that differs from the ones in the healthy cells. And you will find also evidence of fragments around in the blood, depending on the status of your cancer mass growth. And this is our ultimate goal. So this is the final kind of data we would like to be able to use. But we started from this motivation to develop tools that are able to use a mixture of samples and mixture of different signals from one particular individual. So before getting into the details, I will need you to get acquainted with some biology, really simplified notions. First of all, we need to talk a bit about genetic variations, in a sense that when we think about human DNA, what happens is that the sequence of letters that comprises the DNA is almost identical in every one of us. We have some differences, and we can talk about these differences in terms of mutations. Some are inherited from parents and completely that characterizes our appearance, for example. Others are called somatic in that they are acquired during their lifetime. And most of them can be, some of them can be natural, like you acquire mutations with your aging process. Others are related to certain diseases. The other concept is about that we need to know is individual inheritance. I already mentioned, and I guess we all know that we inherit half of our genetic materials from mom and one from dad, other half from dad. And again, we are all pretty much similar. So also what you inherited from mom and dad will look pretty much similar most of the time, and there will be some difference in some locations. When you observe 2 different letters in a certain position, we will talk about allele. We'll call these letters alleles. So these are different versions of the same mutation. And since still we have 2 sources, we can observe most 2 alleles. This is important in When we have something I would say more DNA observations, so we start to have a population and some data, and when this will become clear later, but this means that we can talk about averages, or something that I call expected allele frequency, and we should expect this frequency to be around 0.5. And here I want to give some context about the type of explaining why I'm saying that we have multiple DNA observations. So let's try to still think about the fact that we have one individual and we get multiple observations for this individual DNA. And the fact that we are able to get multiple observations for one person derives from how the sequencing data is usually collected. This comes from a process that takes the DNA, unravel it, and cut it into pieces that are multiplied, duplicated, copied multiple times, let's say. And at the end of the day, we come up with these sets of fragments that are called reads, that are all mixed up together. And we need to put those back in order. And to do so, we use something called the reference genome, which is not the genome of the individual we're considering, but rather like the genome of one standard human being. And once we put this back in order, we have this kind of situation here. We can look at different things in this kind of data. And we're going to look at one particular piece of information. We're going to look at each position in the reference in the genome. We're going to look at the letters for each of the reads, and check whether it is the same as the reference or not. If it's the same, we're going to call it reference genome. If it's different, we're going to call it reference allele. If it's different, we're going to call it B allele or alternative allele. And we're going to keep the counts. So we're going to keep track of the counts of the B alleles and the number of reads. And our first aim with this data from one individual is to estimate what is expected allele frequency. Just to give a peek of the data, here I plotted some raw measurements. So we can take the B allele frequencies and divide it by the number of reads. So it's basically a proportion. And here I plot some data for chromosome 9 to 13. And this is what we can look, these B allele frequencies will look like. And the idea is that I need to estimate the average of these dots all over the genome. To do so, we can rely on assumption that this average will be a piecewise linear function of our data. We don't estimate, I mean, there are some change points where this mean changes, but we are not going to estimate those, but we will be given these points. So that's what it means here when I say that the data is divided into segments. And here, the first things you may think about doing is to take the average of this point for each segment that we are given. And this actually doesn't really work. You will need to believe me on this, but the real doesn't work because we have a problem with the labeling, in the sense that my final target parameter, the allele frequency, is defined in terms of inheritance, is defined in terms of alleles that come from mom and alleles that come from dad. And the labeling that I have been using from the sequencing data, reference and B alleles, does not reflect these processes. So we're going to have biased estimate most of the time. So here I'm in the situation that if I were to know the parental information, I would be able to come up with this sort of plots here. So I will be able to associate a color to each of my B allele frequencies and estimate the right average for these segments. Well, if I use B and reference alleles, what happens, for example, in this case here, instead of getting this estimate around 0.8, I would get something more closer to 0.5. So we don't have access to the parental information, but we have access to some proxy information that is called haplotype. It's not precisely, doesn't precisely mean that we have the parents, but it's pretty much correlated. Let's assume that is almost the parent. It's going to be easier for the rest of the talk. So we have, for each locus, for each genomic position, we have an estimate of this mom or dad inheritance. And of course, this is going to be affected by some uncertainty. So we're going to use this as a prime information in our modeling framework. And this will make up for aim number one. In a sense, this will help us to correctly estimate the allele frequency. Second aim is to be able to deal with the samples that are a mixture between signals from healthy and tumor cells. And here I put together some data from an experiment where some scientists took fragments from healthy cells and a tumor cell and mixed it together in different proportions. As you can see, we go from healthy to tumor cells 100%, and what happens is that for some regions we have that the dots goes towards the edges of 0.1s, and some stays around 0.5. So our second aim for this modeling framework is to be able to distinguish between these kind of situations. And when we have segments that are significantly different from the expected frequency of 0.5, we will call this situation allelic imbalance. Okay, we needed all this to finally put some notation for our model. So here we have a set of observed quantities, the number of alleles, the number of segments, that are the number of reads, and these are classified according to some segments, and an estimate of the parental haplotype. We have some non-observed quantities, which is the allele frequency for each segment, a true parental haplotype, which may be different from the one estimated, and something that accounts for this difference, what we call like a switching error. Switching errors, we know that those have a serial dependence, so we're going to use a hidden Markov model to model all these quantities. Here, I think that if you've been using hidden Markov model, this scheme should be pretty easy to read, but if you don't know anything about that, I will have just use a simple example. Here, when I talk about emission distribution, this is the distribution I'm assuming for the data, and this is we take in account the number of reads and the number of alleles. It will be indexed by a parameter that we call latent state, previously been called switching error, and depending on the value of the switching error, we will have different distribution. So here I took an example where I assume that we have one position, and we have information that for this genomic position we have the number of alleles is actually the number of alleles from that, and we have two different distributions depending on the value of the latent states. In the first case, our estimate was correct, so we have a binomial index by theta det. In the second case, we need to switch our estimate and use a binomial index by theta mam. And since we have just two possible outcomes, we can just reparameterize this in a simple way. I mean, this is a really simple model, but it's just to tell you that at the end of the day, we just need to estimate one parameter for each, regardless of which value the latent states we are in. And we are going to assume that there is some dependence between this value of the latent states. So we're going to, this dependence is going to be Markovian. And the transition matrix is something that tells us with which probability we stay in the latent state equal to 0, and in which probability we're going to change. And these take into account of estimating correctly, I mean, to correct the labels of our points, to correctly estimate the average, the allele frequencies. Finally, to distinguish between situation of allelic imbalance, we decided to use spike as lab priors for our allele proportions. And in a nutshell, we're going to assume that our allele proportions come from a mixture of two distributions, where one distribution is concentrated around 0.5. So this is like summarizing the hypothesis that there is balance in that segment. And the other distribution is probably flatter than what I draw here, but it's going to account for situations where there is no balance. And at the end of the day, this gives us a posterior probability for each segment to be in allelic imbalance. So we're going to use, quickly, I'm using a Bayesian framework, so I'm estimating everything via MSMC using a software called Nimble. And here, quickly, some results. Here is plotting some results from the experiments with the mixture of fragments from tumor and healthy cells. Here I reverse the dots according to the estimate of the haplotype. And the blue line shows the posterior allele frequencies with the credible intervals, and the gold, the lines, the probability of being in imbalance. And I mean, for high signals, we get fairly good results, and we're pretty happy we're able to still get some signals when the tumor fraction is really low. And these are still preliminary results. There's still a lot of work we have been doing, but I think I will stop here and leave time for questions in case there are any. And thank you all.
Thank you very much, Sally. I love this talk. I love the simple explanation of the genome and alleles. I don't see any question from the Q&A right now, so I want to check if there's any question among the speakers, among the other panelists. Oh, I do see a raised hand. Alejandra, if you can write your question in the chat, I don't think I can allow you to speak. So can you write it in the Q&A?
In case I can address it writing if you need to go on with the other speakers.
Okay. Yes, maybe let's go on so we have plenty of time. So Ale, if you can please write it, then Sally will reply in the chat. So now we have Margherita Grushanina. Margherita recently completed her PhD in econometrics and statistics in the Vienna University of Economics and Business. Her research focuses on developing Bayesian methods for unsupervised data analysis of complex data sets. Her interests also include mixture models, Bayesian latent factor models, Bayesian non-parametrics, and hierarchical modeling. Margherita, please, the stage is yours. Okay, we are not seeing your slides though. We're seeing the.
Sorry, this is not what. Sorry, I don't know what is with my computer today. I'm so sorry for this.
No worries, that's fine.
Yeah, mix up. Somehow it doesn't work how I wanted it to.
So maybe try stopping sharing and try share again and see if you can share, I don't know, PowerPoint or whatever it's.
Do you see now the slides?
No, we're seeing the Zoom page, the Safari page.
Okay.
Maybe if you can try stop sharing.
Yeah, I'm
share again.
Somehow, I don't have the option stop sharing. I have new share and resume share.
Oh, I see.
How about now? No?
No, still no. Because I think it's just recording. Oh, yes, now yes.
Okay, perfect.
Perfect.
Perfect. Thank you very much, Cecilia, for the introduction. I will then start with the presentation immediately. We had, as a motivation for this work, we had a data set of inflation rates for 19 Eurozone countries for the time period between February 1997 and October 2019. So 273 observations in total, and we can see that basically we see that the inflation rates behave differently during this time period. So in the first half, there's quite a lot of variation, and the second half, there's less variation. So we can assume that maybe there are at least two groups, two time periods in this data, or maybe more. And if you want to know what underlines, which features underline this data set, then probably we can say that maybe there is a wine driving force in the second half and maybe something more going on in the first half. So if we do any kind of normality test for this data set, we see that most of these inflation rates, they actually don't follow the Gaussian distribution. So this data set shows quite a bit of non-Gaussianity. So we were thinking what methods to use to analyze this data and just in general such data. And one of these methods, it's a mixture of factor analysis model. When we can see, see if there are different groups in the data, so different clusters, and in each of these different clusters, there can be different factors which are driving forces of the data. So this model basically represents a mixture with some weight at each cluster. Each cluster is characterized by a normal distribution, and this normal distribution has some mean, and the covariance of this distribution actually can be represented as a covariance of the factor model. So in each cluster, we have a separate factor model, and of course, we want to assume that in each of these clusters, the number of factors can be different. In the literature, there are such models which allow to automatically derive the number of clusters in such data sets and also automatically derive various different number of factors in these data sets. Usually such models assume a non-parametric prior on the weights of these mixture models of this of the components in the mixture. And this assumes that the number of clusters can potentially go to infinity. Similar idea is employed for the factor models also to derive the number of factors in the data set. So it is assumed that lambda, the loading matrix, has infinitely many columns, and a shrinkage prior is assigned to the elements of these columns, and the shrinkage is done in such a way that the elements are decreasing in magnitude as the column index grows. So in the current literature, some examples of these priors can be multiplicative gamma process prior or cumulative shrinkage process prior, and usually an adaptive Gibbs sampler is used to actually for the inference on the number of clusters, number of factors. And here there are examples of these priors. So multiplicative gamma process prior, we assign a normal prior to the elements of the factor loading matrix, and the precision of this prior is basically divided into two parts. Phi represents the local shrinkage, so the shrinkage of every element in the column, and tau represents the column-wise shrinkage. So basically it is responsible for the decision if we include this factor or we discard this factor. And this tau represents actually a product of gamma distribution, so delta with the column index L, it follows a gamma prior, and of course, as the age, so the column index grows, then we have more of these gamma priors, and the magnitude of this tau will be less. And the cumulative shrinkage process prior, it's in a way similar, so that we assign a normal distribution to the elements of the factor loading matrix, but each variance in the column is assigned a spike and slab prior, where spike actually is some small value like 0.05, and slab here follows the inverse Gaussian distribution, but can be actually any continuous distribution. And slab represents active factors, and spike represents inactive factors. So how the adaptation of the factor dimensionality is done, that in some step, at some iteration, if we use the MGP prior multiplicative gamma process prior, then we have to choose some neighborhood of 0, epsilon, and we see if enough elements in the column in magnitude they're less than this epsilon, then the factor is discarded and all the its parameters are discarded. If there is no such a factor, then a new factor is generated from the prior. And if the cumulative shrinkage prior is used, then inactive factors are identified as the factors assigned to the spike and they're discarded. And if there are no such factors as a certain iteration, then a new factor is generated. However, this model has some drawbacks as well. First of all, these adaptive Gibbs samplers, they are quite difficult to converge. And also programming them, it's a bit of an issue because it's quite cumbersome when model changes dimension at every iteration. So it would be nice if one could simplify that. Also, when we discard the factor and also all the loading matrices at some iteration, then we lose some information because usually these loadings are not exactly 0. There's some noise around them, and it can introduce some bias when we want to estimate covariance matrices. Also, we have to choose some truncation criteria, like these neighborhoods of 0. Also, for the adaptation, we have to also choose some parameters of the adaptation for the probability which is necessary for convergence. And this criteria is quite subjective and actually quite influential. So we need some clear guidance rules on how to choose these truncation parameters. And lastly, if we want to have the econometric identification of the factor loading matrices and idiosyncratic noise covariance, then for infinite factor models, it also remains an issue because these identification rules, they assume certain limit on the number of factors, and it's not clear how to actually hold it for the infinite factor models. So the idea which we had was to actually keep the number of clusters k and the number of factors hk finite at all times, but still maintain the automatic inference on these both parameters. So how we did it? We decided to replace the non-parametric prior on the cluster weights with the dynamic mixture of finite mixture model, which actually represents a mixture model with a prior on the number of components k. And then the mixture weights are assigned with a multinomial Dirichlet prior, which depends on this number of components. And if the prior on k, on the number of components in the mixture, put all its mass on the infinity, then we have the Dirichlet process mixture, and we have this connection with the non-parametric prior. So with this model, both the number of components and the number of field components, which represents actually the number of clusters in our data set, they're both finite and both random a priori. And in the factor model, we replace the non-parametric prior on the factor loading matrices, which so-called exchangeable shrinkage process prior, which actually is kind of a representation of the cumulative shrinkage prior. Just the cumulative shrinkage prior is a limited case of it when the number of factors goes to infinity, and for the finite sequence, we can use this exchangeable shrinkage process prior. And so we can assume some fixed number of factors h, which is the maximum number of factors we can have in the data set. And this number will be fixed for all clusters. And our active factors, the number of active factors will be less than h for all k. So we can keep the fixed parameter, the fixed number of factors for each cluster, and then we don't need to change in dimension matrices, and we don't need to actually discard any parameters. And then we introduce some latent binary indicators for each column on the factor loading matrix so that we can classify the column into active and inactive. So here there's a bit more details on these both priors. So the dynamic mixture of finite mixtures, it's just represents basically finite mixture models where we put some prior on the number of components. And in our case, also a difference will be that the covariance matrix in the normal distribution of these mixtures will actually be represented as the covariance matrix of the factor model. And the dynamic in the name, it actually describes the fact that the Dirichlet parameter, it decreases linearly with the k. So it depends on the k, on the number of components. And the prior which we use to derive the number of factors, we assign the normal prior to the elements of the factor loading matrix, and then we assign a spike and slab prior, almost exactly like in the case of the cumulative shrinkage process prior. However, we have the probability tau of the slab, not of the spike, and 1 minus tau is probability of the spike, and we have the first shape parameter of the beta distribution as our concentration parameter divided by the number of factors. And so basically, the difference is that in the case of the CUSP, we have the basically ordered spike. We have an increase in order of pi here, increase in order of pi, which is responsible for the spike. And in our case, we have actually an unordered distribution, but we can order tau in the decreasing order, and then we have the cumulative shrinkage prior representation. So by integrated out thetas, we can represent our elements of factor loading matrix as actually a sum of a mixture of t distributions. And for the MCMC estimation, we can introduce a latent binary indicator hk for each column, which shows which is 0 if this factor belongs to spike, and is 1 if it belongs to the slab. And in this way, we can do classification of our columns into active and inactive. So the inference can be done in the Gibbs sampler Just the difference will be that we have kind of a nested Gibbs sampler. In the first block, we update the partition. Then in the second block, for each field cluster, we run a separate factor analytical model. So for each cluster, we fully run the Gibbs sampler for the factor model and derive the number of active factors, and then we update the hyperparameters of cluster-specific factor models, and then we come back to our mixture model and sample the new number of components and Dirichlet concentration parameter. And then in the fourth block, we compare our new sampled value of the components in the mixture, and if it's bigger than the number of field clusters, then we generate empty clusters and fill in the cluster-specific factor models in these empty clusters from the priors. So this model works quite well, and this is what I want to illustrate on some examples. So we did a simulation study. The first one is quite a simple simulation study with 3 clusters, all with equal weights. Each cluster has a fixed number of 4 factors in it, and the priors are quite standard. There's a standard normal prior with isotropic variances for the factors, inverse gamma prior for the variances in the factor models, and also just the normal prior for the elements of the factor loading matrix. And then we mixed a little bit the means, so that the factors are not very well separated. Here there are just randomly chosen 5 variables, and then we can see that the classes are quite mixed. And so here there are the results of running 5 times, running our sampler, generating this data set 5 times and running our Gibbs sampler on it for 3 different dimensions. So first case is we have 10 variables and 100 observations, and then 30 variables and 200 observations, and 50 variables and 500 observations. And we can see that the model has done quite well. In red, there are actually the errors where the number of factors was a little bit underestimated or overestimated. And only in one case, one observation was actually misplaced within the clusters. So we had the second simulation.
Margarita, sorry if I interrupt you. I think to leave space for the others, if you can wrap up in a couple of seconds.
Okay.
Yes.
Then I'll leave the second simulation study and just show the example on the inflation data set, which I showed in the beginning. So when we run our model, there are 6 clusters in this inflation data set with either 3 or 2 factors in each cluster. And when we run the alternative model with the non-parametric priors, which I described in the beginning, there were 20 clusters, and basically each cluster was more or less like 1 year in the data set. And here we also try to explain these clusters, so they actually quite fit. If we see what happened in these data frames, so the first period was the period before the introduction of the euro. The second one, when the euro was already introduced. Then the third period, the green one, was with the extension of the eurozone. Then the blue corresponds to the financial crisis. The dark blue to the European sovereign debt crisis, and the last one is the low inflation period with the loose monetary policy. And then it also is interesting to see that somehow in the period of turbulence or crisis, there were usually 3 factors, and there were 2 factors in the more calmer period, so which might represent some other forces which were in play during the crisis periods. So I will wrap up on this.
Thank you. Yes, sorry, I really need to we need to go on with another speaker. Thank you so much for the talk. This was really good. So if there are any questions, I ask you if you can write them in the chat. Rita will be able to answer them in the chat. If you can allow Maria Mamajiwala to share her screen, I'm going to start introducing her. Maria is a research associate in the Department of Computing Science at the University of Sheffield. She also serves as an affiliate researcher at King's College London. Her research has very different subjects, including non-convex optimization, Markov chain Monte Carlo, stochastic optimal control, and system identification via data assimilation. Maria, I'm very excited to hear about your talk. Your stage is yours. Yes, we can hear you.
Thank you, Cecilia, for the introduction, and thank you to the organizers also for the invitation. So I'm going to talk a little bit about my work on data assimilation, specifically in the context of estimating parameters. This is also known as system identification in certain fields. So let me start by defining the problem and setting up some notations. We have a system given whose dynamics is given by this stochastic differential equation. X are the states and thetas are the parameters. And if the theta was known, this would just be a constant, a known constant. But in problems where this is not known, these are taken as additional states that evolve as Brownian motion. So this equation here. And this is important because this is where we propose a modification. So now this becomes like a combined state and parameter estimation problem. In addition to the process dynamics, you are given a measurement dynamics, which is how the measurements relate to the states of the system through this function h, and they are noisy, and they may be partial. You may not have all the states, measurements for all the states. And finally, we have the actual measurements observed at certain time instance. So the problem then is to determine the posterior of x and theta combined, given this set of measurements. And without any loss of generality, we are working with the ensemble Kalman filter. This works in two steps. The first is you start with an initial ensemble for the states and parameters and pass them through these process dynamics and parameter dynamics. So this is the prediction represented by a tilde. And the next step, which is the actual data assimilation step or filtering step, is obtained by giving an additive correction to the predicted ensemble. Here's the z minus h of x term. This is known as innovation because this is where the new information comes in through the observations z. And h of x tilde is something like a predicted observation. So if you had perfect states, you would expect this to be a zero mean error. K here is the Kalman gain matrix, and it's a function of the states, the parameters, predicted observations, and this measurement noise intensity. It's a straightforward formula to determine a matrix. Now, mathematically, this is perfectly okay to use Brownian motion for the evolution of parameters. However, in practice, you face certain issues. I'm not able to. Right. So there are certain issues while implementing that in practice. There could be no convergence, or the convergence could be slow, especially for the parameters. Then the sampling variance could be large, which means if you run the algorithm over and over, the results would vary a lot for the converged parameter values. And also it becomes increasingly difficult with an increase in the problem dimension. And finally, here we observe because parameters don't really have any physics associated with them, the measurements are only used indirectly to update the parameter values in the update step of filtering. So motivated by these issues, we propose a modified dynamics by introducing a drift in the parameter dynamics, which directly uses the measurements. So I come to the main idea. The idea is to construct a pseudo energy using the measurements, and then use this energy to construct a Langevin diffusion equation. So assuming I knew what this E was, this is what the parameter dynamics would look like. Here, beta is a temperature-like quantity, and it relates to mobility. So it should be high initially, so we explore more, and then slowly reduced as we start converging. The other two equations remain as it is, so the process dynamics and the measurement dynamics remain unchanged. And these three equations now define the problem. So basically what we are doing is we are just changing how the problem is posed. And so once we have these set of equations, we can use any filtering algorithm, not just ensemble Kalman filter. All right, so the question first is how to define the energy E. There are infinite ways to do that, but certain points must be kept in mind. So because it's an energy-like quantity, it should be non-negative. And ideally, it should be unimodal with a minimum at the desired location. So the one possible choice that we're working with is this expression. Z minus h of x is the same term that is used in the update step of filtering alongside the Kalman gain. So this is just a choice. It could have been just the square as well. So this is an error squared like quantity, an exponential of that. So if I call this term inside the exponential as v, then this is what the gradient of E would look like. It contains this squared error, exponential of squared error. Then it contains term-wise, the component-wise errors appropriately multiplied with the partials of h, which is the measurement function, and then partials of x. So to get the partials of x, we just do an implicit Euler-Maruyama integration according to this equation, and it would just be partials of f. And here the repeated indices mean that it has to be summed over. So if we sum over p's, sum over j's, what we're left it is vector of size equal to the number of unknown parameters. So just for completeness, so for this particular choice of E and this value of gradient of E, this is what the modified parameter dynamics looks like. Right. So coming to the numerical examples, I have one example here for the Lorentz 1963 model. This is a three-state, three-parameter system. So the x, y, z are states, sigma, r, b are the parameters. And this is a chaotic system. And this is what a typical response for this system looks like. It looks like a butterfly and also exhibits the butterfly effect, which means if you like a small change in the initial conditions can cause large changes later on. So these type of systems are particularly more difficult to calibrate. Working with these true parameter values, so this is a toy problem. I start with these initial conditions and generate a reference solution, add noise to that reference solution and call that as the measurement. So I'm using a measurement noise intensity of 2, which is quite high. And then use the two methods, the original parameter dynamics and the modified dynamics, and compare the results. So this is the intensity for the original parameter dynamics, noise intensity. Right, so these are results for the posterior of the x and the xyz states and the parameters for an assimilation frequency of 1, which means that we have a measurement available at every time step. So every time there's a prediction, we also have a measurement available to assimilate. On the left are the parameters, and on the right are the states. And so the pink plot, so if I look at one of these, the pink plot is the one with the modified parameter dynamics. And the number here represents the ensemble size used in the ensemble Kalman filter. So for an ensemble size of 10, the pink is a solution with the modified dynamics, and it should be compared with green, which is the original parameter dynamics, that is Brownian motion. And just out of curiosity, I also have two more plots here, the dotted orange and blue, which is for the original dynamics, but with a larger ensemble size. So with 100 and 1,000. So I just wanted to see if I increase an ensemble size, can that still compare to the modified dynamics? And as we can see, even with a size of 1,000, it doesn't compare very well, at least for one of the parameters. And because the states and parameters are coupled, when the convergence for parameters improves, automatically the convergence for states improves as well. So the next study I did was for sampling variance, which means running the same algorithm over and over. So this is done 100 times, the same problem that I showed earlier. The same problem is repeated, the algorithms are repeated 100 times for an ensemble size of 25 with an assimilation frequency of 1. And these are only the parameters, because that's what we are mainly interested in. So the left side is the original, which is the Brownian motion. And on the right, we have the modified parameter dynamics. The individual gray lines are the results from an individual run. The red lines are the true parameter values, the reference values. And the black is an average over the gray, over all the independent runs. So as you can see on the left, there's quite a bit of variation. And also there's a small bias. This would probably go away with a larger ensemble size, but it doesn't seem to be an issue with the modified dynamics. Right, so then I did the same example, but with an assimilation frequency of 10, which means we have 10 times less data than before. So we assimilate only every 10th time step. The original dynamics doesn't change, which is a Brownian motion. But if you recall, the modified dynamics depends on the measurements. So until a new observation is available, we keep using the same observation or the same gradient in the Langevin dynamics. So again, the colors are the same. So modified parameter dynamics is in pink compared with the green. And even with a higher ensemble size, we still see that it doesn't compare very well. Same is true for the states, because the behavior of convergence between states and parameters is related. When parameters converge faster, so do the states. Right, so to quickly summarize, what we did was modify the parameter dynamics to introduce a drift, which utilizes information from the observation data, from the process dynamics, from the measurement dynamics. And we observed that the ensemble size required is reduced, and so is the sampling variance, which is sort of more robust. And the overall quality of the results is also enhanced at a small increase in the computational cost. So the increase in computational cost will depend on the problem, but at least for this particular problem, it was about 20% increase in the cost of computation. And as I mentioned earlier, the convergence of states improves as a result of modifying the parameter dynamics as well. Finally, for some future directions. So the first step would be to undertake a convergence analysis. This is quite tricky to do in any data assimilation method, but it would be very important. And then other possibilities are to change the versions of the dynamics that is used. So there are many different versions of even the Langevin diffusion that's available. One of these I developed in my earlier article was a Langevin diffusion on Riemann manifolds that was used in the context of optimization, but it could readily applied here. Then we could also use generalized Langevin dynamics. So this is something that uses history dependence, and it could be useful when the data available is limited. And finally, if the parameter dimension is very large, we could switch to stochastic gradient Langevin dynamics or one of its variants. We could also play around with this form of the energy, the pseudo energy that's being used. And that's it. Thank you very much for listening.
Thank you very much, Maria. Thank you for your great talk. I will ask to write any question in the Q&A session, so we can have Maria Vladimirova start with her talk. If you want to start to share your screen. Maria is a research assistant at Criteo Lab based in Paris and Grenoble. Her research mostly focuses on explaining and improving machine learning methods with instruments of Bayesian and causal statistics. Recent work includes how to connect Bayesian and standard neural networks, fairness assessments, and in recommendation systems. And Maria, the stage is yours.
Thank you very much. And thank you for the introduction and for inviting me to this talk. So I'll try to make it on time. So I was working mostly on the Bayesian neural networks. And here I will present how we can help to get some understanding into some models like neural networks, very complex models that can use very complex data and get very meaningful information from this data. And with the Bayesian approaches, we can get some understanding how they do this, or at least help us to get more insights. For this, I will start with describing what is the neural network, what is Bayesian neural network, and then I will provide some properties of these neural networks, more precisely about their infinite width properties and then finite width properties, which I studied in my research, and then I conclude with the insights and with the future works. So let's start with the Bayesian neural networks. For this, firstly, we need to describe what is a standard neural network. So this is a hierarchical model made of layers. We have an input layer, and usually it's you can separate any input into the features. And then we apply a linear combination of the features with the coefficients W, which are called the weights. And this linear combination will be an input of the layer. Then we apply a nonlinear function, which is also called an activation function. And the output of this activation function is the output of the hidden unit of the layer. And we do like this, h capital times, and this would be the number of hidden units in a hidden layer. Like this, we have L hidden layers, which is the depth of neural network. And in the end, we have an output. And for this neural network, we need to learn weights. So we need to get a training algorithm to learn weights. And in the end, when we get an object, which is X with the features, and then we get an output. And this is called the forward pass. And to update the weights is called the backward pass. So to choose a neural network, we need to choose an architecture. Here I described as standard linear layers, though they can be several, such as convolutional neural networks and so on. And we all heard about ChatGPT. So it's just a complex layers of this very simplified neural network that I just described. And then we need to choose a training procedure, how to update the weights. So for example, gradient descent, stochastic gradient descent, and all their differences. And then these models, they have great properties, such as they're very expressible. They have good adaptive bias and very good generalization ability, though they have limitations, such as transparency. It's a black box. So we give an object and we get something that we don't know how it worked, but it did. And then it doesn't provide uncertainty estimates, and therefore we cannot really interpret it. And so here, there comes Bayesian that can help with providing uncertainties and therefore interpretability. And for Bayesian neural networks, what we do is instead of point estimates for the weights, we have a prior. And then when we propagate, so we get linear combinations, the output will be also random variables. So it will be some distributions. And then the output of the whole neural network would be a random variable. And so the problem then, which it comes to us, is how to update the weights. This is through the, we need to get the posterior approximations. So, and with these models, in the end, we get the uncertainty and then we get interpretability. And then the question is how to get the approximation, though we can help into answering the questions of transparency. And maybe it can help to use the standard neural networks. We have good training procedure to get the weights, though we will help with the questions of transparency. So we get some insights. And to do this, we get the properties. So some years ago, there was the work done on the infinite with the neural networks. So it's when the number of hidden units tends to infinity. And then it was shown that in this, 1996, it was shown that actually one hidden layer is equivalent to a Gaussian process, which is a well-known model. Everybody knows it, and everybody knows how to get a posterior. It was a breakthrough, though one hidden layer doesn't bring us much gain. We need many more layers, and there it was not clear what we have. Though in 2018, 2 papers show that actually all hidden layers, if we turn them to infinity, which is called infinite width, we get the Gaussian process. And this show that actually standard neural networks, when we apply prior, when they get a prior, then the equivalent to Gaussian distributions at the hidden layers. So, and this brings a lot of research further, because we know a lot about Gaussian processes. So it was shown that we can get posterior approximations, we can get neural tangent kernel, we can apply some initialization at a standard neural network, which can be transformed into a Bayesian neural network, and to propagate information further. So this research helped to understand what we should do with a standard neural network. Though we actually have finite width, so it's not perfectly correct to assume that we have infinite with neural networks. And it was shown that these Gaussian processes at the limit, they have poorer performance than the Bayesian neural networks with some inference. And they have lack of representation because in the end, Gaussian processes, they have a fixed kernel. And they work worse than Bayesian neural networks. And then because of this as well, transfer learning is not really possible. So our goal was to study finite neural networks, because even if we consider distributions of the hidden layers, so going deeper, they are further and further from Gaussian processes. If we take, for example, 50 hidden units at the third layer, they're already not that close. So, and then this was where my research comes for the finite with neural networks. What we studied, we came to this into three parts, kind of, three sides. We studied the dependence, because in this case, in the limit, we don't have independence. And this was shown that for the infinite with the neural networks, we have independence. Then we studied the shapes of the distributions, and we showed some tail characteristics. How we did it? So consider the distribution. We have some center and we have some tail. And the center we study through the concentration inequalities, and the tail through some tail characteristics. So, and since the new hidden unit in the neural networks, it's a linear combination of the units of the previous layer. So then these hidden units in the one layer, they're dependent between each other, because linear combinations of the same hidden units of the previous layer. So, and this makes everything complicated. And then we need to some dependence properties. And for example, consider this dependence function, which has easy interpretation, that it's the difference between the joint distribution and the product of the marginals. And so if it equals to 0, then it's independent. If it's positive, we have positive dependence. If it's negative, we have negative dependence. And so we checked that actually in neural networks, there are some interesting properties. If we go deeper, we have kind of more dependence in some cases. And we have, if we go wider, we have kind of different dependence. And it's possible to describe it, though we have indeed the dependence in the limit. And there are ongoing works on how to describe it better, for example, through copulus or through some different criteria for exchangeable random variables. And we got some descriptions that can help us to get the tail characteristics. And so we got that the tails are actually going heavier if we go wide, if we go deeper. And heavier means that we have upper bounds and lower bounds of distribution heavier than Gaussian, and then the next layer heavier than exponential, and so on. And so in the end,
Just
it's completely different from the infinite with neural networks. And then it's a future work where it starts, where how different it is, and where we have the center and when we have a tail. And this is the reason why we have poor performance for infinite with neural networks. And this can help to get some future works that you can see on the slide. And thank you very much for your attention.
Thank you very much, Maria. Sorry for not leaving a lot of time. This was really, really interesting. Okay, so we don't have much time for question. I am just going to thank everyone for attending this talk. Thank to all the panelists for giving this really great talk. Thank you for the organizers, and enjoy the rest of the event today. So
And if people have questions, please reach out to the speakers. I know there were some questions on the Q&A box, so we will keep collating those questions too and reach out to the speakers. Okay, so please reach out to the speakers if there are questions. Okay, I will stop recording now then.